Hello, everybody. I'm back. Dr. K.K. Agarwal, President, Heart Care Foundation of India, Confederation of Medical Association of Asia and Oceania, and past National President, Indian Medical Association. And you are watching Med Talks with Dr. K.K. Agarwal, 7 p.m. show, 159th non-stop edition. Let's have the headlines of the day. The South Africa Ministry of Health rejects the claim that its COVID variant more dangerous than the UK strain. The question is not dangerous, but contagious. That will also be a subject of discussion today. COVID-19, India's active case hold of 2.8 lakhs now, only 2.78% of total cases. A dry run for vaccine administration will be conducted in Andhra Pradesh, Assam, Gujarat, and Punjab next week. It makes sense for the Modi government to launch the vaccine within this year. We do expect it coming out as a New Year's gift to the people. Several states in the country on alert as 18 more UK flights test positive for coronavirus. I personally feel that the disease in the UK has started from September and December, and all the places in the world where there is a second peak may be the new strain of the virus. The tourism department has issued guidelines for amusement and entertainment parks stating that all customers will have to be screened at the entrance and wear masks all the time inside the premises. The Med Talks journalist, when went to the railway station, found no screening. When railway station is not strict in the screening, the time will tell us what will happen in the amusement parks. Before we go to the subject of the day and announce the subject of the day, let's have the ritual of washing our hands. So we take a extra ultra protection gel, we take it on our hand and we say S for Seedha, U for Ulta, M for Mutti, A for Angutha, N for Nakhun, and K for Kalai. And now we are ready to accept our gift as a ritual. And let's have the topic of the day. As discussed in the past, that this week till Sunday night, we will only be covering more and more about the coronavirus and its mutations. The basic idea of discussing that is that once we know that India is on the top countries list as far as corona management of mortality, maybe the first country to develop herd immunity, our students at a very basic level, MBBS, BSc, why only BSc, even 10th class, 11th class, 12th class, should know much more about virology, cell biology, mutations. They should talk about the genomics. I feel that we are going back to our Vedic philosophy. As is the microcosm, so is the macrocosm. And we need to understand more and more about cellular health. To talk about this virus cellular health, I have a special guest with me, Dr. Pavitra Venkatagopalan. Before I introduce her, let me congratulate her for completing her family, for the baby boy, because first time when I interacted with her, she was at the verge of delivering, and she said that if I don't get a labor pain, I'm going to give the lecture. And today she says, if my baby boy doesn't cry for the milk, I'll continue my lecture. Thank you and congratulations, Pavitra ji. She worked as a coronavirus assembly for her doctoral dissertation at Arizona State University. She heads her own NABH certified diagnostic center in Chennai and Bangalore. With the recent emergence of COVID-19 infection, she has seen many myths in the form of WhatsApp forwards. She has since then talking at various venues to make everyone understand the basics of coronavirus infection and its outbreak. Her talks include dispelling myths, establishing facts, and clarifying doubts from the audience. She has also been addressing corporates in various industries on scientific methods to protect themselves against the infection. She has been invited by the Tamil Nadu Science Forum on National Science Day at the Anna Centenary Library to give the C.V. Raman lecture. Congratulations again. Moreover, she has appeared on various well-reputed shows to talk about current scenario of COVID. She has recently been the recipient of Healthcare Heroes Award in the Mythbusters category by Mr. Health India and Jagran Media Group. So, all to you, Pavitra, to give a presentation before we interact. Only if the time permits, we will call few of the audience who are watching our program to come and interact with you.
Absolutely. Thank you, Dr. Agarwal, for the wonderful introduction. I'm going to go ahead and share a short presentation about what is happening around us in the coronavirus mutation world. So please bear with me so that all of us can be on the same page when we talk about understanding these mutations. One of the first things we need to know about life is we have archaea, bacteria, archaea, which are single-celled organisms, and eukarya, which are multi-celled organisms, and we have viruses. The common thing between all these forms is they have either DNA or RNA, and then this DNA or RNA is the coding language for the protein. To understand it better, the DNA contains a lot more information, which sort of is like an architect's rendering when you build a house. But the technical drawing, like the electrical drawing, plumbing drawing, is the mRNA. And the contractor reads the electrical drawing or plumbing drawing and builds the house, which is the protein. In coronaviruses, there is no DNA, there is just the RNA, which makes the protein. But the common point to remember is they have DNA or RNA. So, like I said, DNA and RNA are these molecules that code for life. The molecule on the side, right-hand side is DNA, and the molecule on the left-hand side is RNA. If you look closely, DNA has two strands and RNA has one strand. However, the base, that is what makes these strands, are the same molecules. Both have cytosine, both have guanine, both have adenine, and RNA has a molecule called uracil and DNA has a molecule called thymine. They are just to distinguish between the two so that the body knows whether it is reading the DNA or the RNA. A combination of these molecules, that is CGAT or CGAU, that gets repeated in various combinations throughout the genome. The human genome has about 3 billion base pairs and the coronavirus genome has 30 kilo base pairs. So if you have, this is your DNA, then it is translated, it's transcribed to RNA. So there is an enzyme called transcription enzyme that converts the DNA to RNA, and then there is another machinery called protein translation. That is, it translates the language of the mRNA for the cell to make the protein. For example, it reads it in chunks of 3. AUG means it one part of the protein. AUC is another part of the protein. So every protein is read through like this in these chunks of 3. Where you start and where you end are all parts of the coding language. It's exactly like how you have Java or HTML software where you say there is a colon means something. This particular stop means it will stop. There are start means it will start. There are specific codons. For example, AUG means that's where the translation machinery will start making the protein. The first protein building molecule is always MET. So it will not randomly start like anywhere. It will not start at UGA. It will not start at GAU. It will search and find AUG and start there. And then based on that, it will move in frames of AUG, AUC, UCG, and so on. And proteins are made up of amino acids. Just like the name suggests, proteins have an amine group and an acid group. And then there are about 22 amino acids that are found in the human body, and all of them, if you see, have the amine group and the acid group. I know this might be a lot of biochemistry and chemistry for a lot of you. All I want you to see is the similarity in these, in all of them. Forget about the colors for now. All of them have the same amine group and the acid group here in all these. And then you have these other side, which I didn't talk about. These are called the side chains. These side chains are different for different amino acids. Obviously, some of them, the reason these amino acids have different side chains is that each of these gives it a different structure. If everything was the same, if the entire molecule was the same, it will not be able to form multiple different structures. So here, the side chains are different types. Some are positively charged, some are negatively charged, some have no charge, some have special cases. They have maybe a ring molecule, some have nothing. Some that have a hydrophobic chain, that is, it will not interact with water. Some have large rings. So what happens in a protein is, in some locations, you need to have an amino acid that is that has a negative charge. In some places, you need to have an amino acid that has a ring. You need to have an amino acid that has a very big ring. So all these factors make a difference in how the protein is structured. So all we are looking at and trying to understand is, each amino acid looks different. Sometimes they may belong to the same group. Sometimes if only a positive charge is needed, any of these could come and there will be no change in the protein structure. Sometimes there will be a negative charge, and if you replace it with an amino acid with a positive charge, there can be a change in structure. If you have an amino acid that is very small like this, and if you replace it with an amino acid that has a ring like this, there will be a change in that particular region in the protein. This is very broadly trying to understand what protein structures look like. Oh, this is a bit hazy. I apologize for that. So there are different types of mutations. Mutations occur when there is a change in the DNA sequence. For example, if the original one has TTC and the mRNA reads AAG, the body knows to use the amino acid LYS, lysine. Sometimes you can change it to TTT, the mRNA becomes AAA, but the body still knows to take the same lysine. However, in this TTC, the first amino acid, the first base is replaced to ATC. Then the mRNA becomes UAG. UAG is a stop codon. At that point, if the body sees it and it has a mutation where there is a stop codon, the protein will stop getting synthesized abruptly, even though originally there may have been more protein being made. This is called a truncation. That is, the whole protein is no longer made, only a small portion of the protein is made. Sometimes it can allow for say TTC becomes TCC and becomes AGG. It can put another amino acid in place. Instead of LYS, it puts ARG. Instead of LYS, it can put THR. Depending on what amino acid replaces what amino acid, the protein might function better, the protein might function worse. Sometimes some of these mutations can allow for the cell itself to stop functioning, because if such a mutation occurs in a very, very important protein, the protein, if it doesn't function properly, the cell itself will die. And this is very important to remember, because all these mutations is what allow for evolution. If there is no mutation that happens, we would not have evolved from a single-celled organism to what we are today. So all these mutations slowly, I'm not saying a single mutation is going to cause like a movie like Species. You're going to have small changes. For example, and these mutations help us adapt to our environment. If you look at the world, people around the equatorial region are darker skinned than people in the northern and southern hemisphere, more in the Arctic regions, etc. This is because people in the equatorial region get more sun, so they don't need to have light color skin. The light color skin is what absorbs the sun. Whereas if you live in regions where there is less sunlight, you need your skin to absorb more sunlight. So your body tells you, hey, if you're in the equator region, you can be dark. You don't need a lot of sunlight to be absorbed in your body. So if you're in the Arctic region and northern and southern hemispheres, you would need a lot more sun rays to be absorbed because you have a lot winter that is a lot longer. So whatever sun you get, you need to absorb that sun rays. You need to absorb that better, so your skin is lighter. This is the simplest form of evolution that I'm talking about, that we can all see clearly. But all evolution takes place because of these mutations. Mutations are neither scary nor artificial. This is how life functions. Now, if the mutations are very, very common, that is, if they always occur, then nothing stable will form. Because most of the mutations lead to some of them can be right, some of them can be wrong. You still want to preserve what life you have. So you have the human genome, which is 1 billion base pairs. During this replication, when our cell copies one copy to another, if there are a lot of mistakes, then it will lead to a lot of problems. The cell might not even survive for the most part. So there is a mechanism called proofreading, where every time we write a document, we check to see if there are any mistakes. Let us say we fill a form. If we keep filling the form without proofreading, we can make mistakes and the form is rendered useless. This is exactly how the replication takes place in our cells as well. When the copies of DNA is made, you have an enzyme that goes and checks whether it is copied it correctly or not. It's called faithful replication. In the human genome, the proofreading is excellent. We have 3 billion base pairs. There is only one error for 1 billion base pairs. So the chances of you having a problem with the one cell copying to another is very, very less. By the same theory, when you look at HIV, HIV is one of the cleverest viruses that we have around. The HIV virus, or most viruses actually do not carry a mechanism for proofreading, because viruses are very small, effective, light creatures of survival. Carrying the enzyme that checks for the proofreading is too expensive for it. So it does not have a proofreading activity. So HIV actually has one mistake for every 70 base pairs. So that most of it is wrong. Most of the copying is wrong. Whatever mistakes that allow it to survive is what survives. Coronavirus, however, has a large genome. It's very large for a virus. In fact, it is the largest known RNA virus. It has 30,000 base pairs. Now, in that, the mistake is 1 in 10,000 base pairs. So, which means there is a proofreading activity in coronavirus, and that enzyme checks to make sure that the virus is copying itself correctly. So, based on this, do we expect a lot of mutations in the virus? Not really. So, what happened in this case? Why do we have this new mutant B1.1.7 having 17 mutations? Let me take you through the process. So this is just a schematic to see what the virus looks like. On this is the spike protein. This protein interacts with the cell to allow the virus entry. Upon entry, the virus RNA is copied by using the cellular machinery itself. And this is what a line of the genome looks like. And you have this is not really relevant. This entire part, this is about 15 kb, that is 15,000 base pairs, actually codes for all the enzymes needed for virus replication, proofreading, cellular immunity, etc. So, and then you have the spike protein. So this is where you have the spike protein gene. The rest of it are other genes that we know that have various differing functions that we will discuss as relevant in the rest of this talk. So when the virus attaches to the cell and entry takes place, the virus genome is there. The virus genome can be made into the viral proteins by the cellular machinery. The cell thinks the viral RNA is a cellular RNA and starts making the viral proteins. And once the viral proteins are made, it takes over the system and makes more copies of itself. Like I said in the previous one, translation of ORF1A1B, there is a protein called NSP14. This protein is thought to be responsible for the proofreading activity. So if this protein is present, it checks to make sure not too many mistakes are made while copying the genome and making more copies of the viruses. So when we started, the theory behind why we have so many mutations is as such. At this point, this theory doesn't have a proof. It is only a hypothesis. So remdesivir is a medication that we've all heard of. Remdesivir acts on NSP14, which is the proofreading activity of the virus. So what happens when you suppress the proofreading activity? The original hypothesis was when you suppress the proofreading activity, the virus will make multiple mistakes, and then as it becomes weaker and weaker, the body's immune system will attack it, and thus in a two-pronged approach, the virus can be defeated by the body. It's an excellent theory. Nobody expected this side effect though. What possibly could have happened is when the remdesivir was given to a patient, the proofreading activity no longer worked and a lot of mutations came into place. Ideally, at this point, the virus should have died. But it may have gone to a patient who was immunocompromised, where their immune system was not killing the virus, and a lot of these viruses with mutations escaped from this patient and continued getting transmitted. So it's a combination of two things. One is the lack of proofreading activity allowed a lot more mutations to take place, and the fact that the virus could not be killed by the host in whom this happened because they didn't have an immune system allowed it to escape. And it so happened that this mutation gave the virus the ability to spread. So when you look at these kinds of mutations, when you selectively put a pressure on the virus, so on the left can be the different types of mutations that are there. When you put a selective pressure where you're applying some sort of a force, either the body's immune system or a medicine, those mutations that allow it to survive will be the largest known population. In this case, that's what this D1.1.7 has been. So over time, the mutations that allow the virus the best chance of survival grow in number and remain. So all the mutations that did not allow this virus to survive very efficiently will die. That's really what happened. And if you notice, okay, I'll go back to this point later. So what is this new mutant? It is a variant. It is not a new virus. It is not a new strain. This is called VUI, Variant Under Investigation, 2020. In the year 2020, in the year December, and variant number 1. It's not just found in the UK, it's found in Australia, Iceland, Denmark, and Netherlands. And it has 17 identified mutations. Not all these mutations are thought to be significant. Now, we know that this virus spreads faster, but not more lethal. Now, this is not something we need to take lightly, because if it spreads faster, it means it's spreading to more people. If it spreads to more people, then more number of cases with serious infection can come. When we have more number of cases with serious infection, you are likely to have more number of deaths. So this is something to be feared. It is not something to be taken lightly. So like I said earlier, it is most likely because of accumulated mutations from an immunocompromised patient. We still don't know if this is true, but this is the strongest sounding hypothesis out there. So I'll be talking about the significant mutations in this particular strain, VUI 2020-12-1. I'm going to be mainly talking about the spike protein. This is a representation of what the spike protein looks like. So you have the protein folded in a 3D structure. So this particular protein actually looks like a club. So when you just open it all out and lay it from end to end, this is N-terminal domain. This is the RBD, which is the receptor binding domain. Like I said, the cell has a receptor to which the virus binds to. The spike protein of the virus binds to the cellular receptor, and this region is very important for that. The RBM, receptor binding motif, is this 6 amino acids, 424 to 494. This is a very critical region for us to keep in mind. So what these numbers refer to are the amino acid numbers. This protein has about 1,300, 1,400 amino acids, and you're looking at different regions that are important. So for example, the most commonly talked about is the deletion of the amino acids 69 and 70. At this point, we don't know what effect it has on the virus in infecting the host cell. The most crucial mutation that is found, N501Y, this is a positively charged amino acid being replaced by an amino acid with a ring. And we know that this is very close to the receptor binding motif, which means it is affecting something to how this spike protein binds to the cell. The theory is that when you do modeling studies, you can test this theory out hypothetically. It is thought to be that the spike protein binds the cellular receptor with a much stronger affinity. What happens when it binds with a much stronger affinity is, even when there is a smaller amount of exposure of this virus to the host, there is a few particles are also enough for the virus to infect the cell. This is probably why this variant is spreading faster among people. And the third critical mutation in this is Y145 deletion. This also lays here. The spike protein is what is seen by the body during the infection. So when the spike protein is seen by the body reacts and makes antibodies to bind to it. These antibodies coat the virus, and the antibodies take the virus to be destroyed. Now, what happens if the antibody cannot see this protein, cannot see the spike protein? Then we have a problem. We'll have to make new antibodies, and that takes time. So this Y145 is known to be in a region where a neutralizing antibody binds. So what will happen if someone is vaccinated and then they get infected by this Y145 deletion strain? We still don't know. The theory there is antibodies are very large molecules that recognize large structures. So a change in one amino acid may not have such a significant difference in how the immune system plays a role in controlling the infection. Again, these are just theories. We still don't know what it means. We'll have to wait and study. So one of the important things that people keep asking me is this the only mutation that comes in the world? It is not. So far we have had about 3,700 genomes being sequenced around the world. So if you go to this website, nextstrain.org, it tells you the different strains that were sequenced from different regions and where all they had the different mutations. So seeing mutations in various parts around the world in various genes is not something very surprising or special. It is something that continues to happen when there is an infection going on or when there is a pandemic. So this, you can see that this will continue. This will tell you how many amino acid changes. You can actually look at these, where are the different changes, what are the different mutations and things like that. If you look at it carefully one by one, I'm just showing it to you so you can go and see it by yourself. It's called nextstrain.org. Now, what does the future hold for us? The future, what we think is the current vaccines are likely to work, because a vaccine is not going to give us just one antibody. It is going to give us multiple antibodies, which should still neutralize the viruses if we get an infection from a slightly different variant. And it is important to remember that because of the genome size of the viruses and the fact that they can carry a lot more mutations than we do, they will always evolve faster than us. So constant surveillance is essential. This is why sampling viruses from patients from different parts of the world at a frequent level is important. In the UK, they sample from around 5 to 10%. I don't know how many patients they sample in India. Maybe Dr. Agarwal can get the answer for that. That being said, what do we do about ourselves? The basics still work. Like Dr. Agarwal started this lecture with hand washing. Effective mask wearing, distancing and hand washing still work. And until we all get the vaccine, this is the best method to keep ourselves safe from COVID virus or any other variants that it may show up, that it may come in the future. Thank you.
Thank you, Pavitra, for a detailed, exhaustive understanding of the virus and its mutations. So again, I will now likely to concentrate on the mechanism.
Yeah.
Normally, we say that virus is dead in 7th, 8th, or 9th day. And then we say the virus may still replicate in immunocompromised for 20 days. And severely incompromised, it has been seen even for 3 months. In cases with severe immunocompression, cancer, and other types of patients. So 3 months replication is known. Now, in such cases, multiple mutations are bound to occur. Yes. So it is very clear that immunosuppression patients misuse of many things can cause mutations. One of the theory for misuse is plasma therapy when it is not required. Now you are talking about remdesivir when it is not required.
It's
So
still a theory.
normally,
We don't know if it is,
no,
that is
no,
the case,
no,
but
no.
it's all the hypothesis.
We are all talking about copy left.
Yeah.
Copy left means take it from here and work on it.
Yeah, exactly.
Copyright means this is a theory under investigation, not a theory of concern.
Right.
As you have a variant under investigation followed by variant of concern,
Right.
it is not yet VOC. So we are not thinking about a theory of concern, means it
Okay.
has been proved. We are still discussing theory under investigation, like it
Right.
is for a variant with that. So it makes sense to concentrate on the work of NSP14. Out of all the non-structural proteins, it makes sense to work on NSP14. What about other antivirals? NSP now, can you tell us a little bit more about NSP14 and other NSP?
So the different non-structural, let me pull up the genome, please.
Yes. Can we understand more about the 1 to 15 NSPs?
So, wait.
Because now my mind says is one should focus on NSP and try to find out diet, exercise, what changes NSP, apart from which drugs, which diet, which plasma,
that,
which antibody. When you have got a focus, that NSP is
yeah.
the focus now. If
So,
yes, can you show us that NSP flight?
so I don't have a figure for NSP. NSP basically means non-structural protein.
Agreed.
So the structural proteins are basically these: spike, envelope, membrane protein, and nucleocapsid protein, meaning they form the structure. They basically are responsible for building the virus. The non-structural proteins are the functional proteins.
So are they 15 or 16? 1 to 15 or 1 to 16? NSP.
NSP,
You
I
wrote
think
about 1 to 15.
No, I have written 1 to 15. It's 1 to 16, but this is from the previous, this is from SARS one. I just took my old slide and put it here. I should have changed this.
So this is 1 to 16.
Yeah, it is 1 to 16.
But every NSP has to have some work.
Yeah. The full functions of all the NSPs are still not known, simply because we just haven't studied them just yet. So we know that all the non-structurals assemble to form the enzymatic activity that is responsible for the virus replication and proofreading. We still don't know some of the functions of some of the different NSPs. So that work still goes on. The NSP14 especially again came into predominance because of this virus, this COVID-19. It is not particularly very interesting to study otherwise.
So which enzyme is responsible for 14?
Which enzyme? No, NSP14 is an enzyme that proofreads the genome when the replication takes place. So, okay, let me go back one step. Okay. No, maybe go forward one step. I apologize. So, in the human body, there is no need for an RNA-dependent RNA polymerase. The purpose of this in the human body has DNA. The DNA becomes RNA and the RNA becomes protein. The human body has only DNA-dependent DNA polymerase because the human genome is made of DNA and copies are made from DNA to DNA. Whereas for this coronavirus, it has an RNA genome. That is, it is an mRNA genome. And it needs to make more copies of itself, which means the enzyme has to look at the RNA genome that is there. It is dependent on that RNA, and it needs an enzyme that can make RNA, more copies of the RNA. That's why it's called the RNA-dependent RNA polymerase. So this is not one single molecule. It is a complex of multiple molecules, which is the original, not the original, the ORF1A, 1B, that forms this complex, allowing copies of the genome to replicate. This is a very interesting target and is a very scientifically sound target for antivirals because this RNA-dependent RNA polymerase has no equivalent in the human body. Because it has no equivalent in the human body, it cannot affect the cellular function. That is why the role of NSP14, the role of the other NSPs are important when it was chosen to attack it for controlling the viral replication.
So NS14 and RDRP are different. They're not same.
They are the same. RDRP is a huge complex.
So NS14, because normally when we talk about is that remdesivir is a RDRP inhibitor. So it tells us that it is RDRP basically is responsible for the proofreading.
Yeah, the RDRP is responsible for making the entire copy. It is responsible for starting the replication. It is responsible for elongating the RNA. It is responsible for checking if the RNA is replicated correctly. That is the proofreading activity. So it is a complex. It's not one molecule. It is multiple molecules that we refer to as RNA-dependent RNA polymerase.
So we are talking about that if RDRP is gone, then it takes away the proofing mechanism. It converts the RNA like that of an influenza, which doesn't have the proofreading mechanism.
No. So we
because
cannot.
In influenza also you don't have the proofreading mechanism. Yes, so it starts working like behaving like multiple mutations of influenza, and it starts doing multiple mutations.
So if you suppress NSP14, which is the proofreading activity of the RDRP, you will have multiple mutations. Influenza is a different case because you can have various other forms of recombination as well. But that will just make it very confusing, so I didn't bring that up here at all.
Okay, so anything we have more about NS, how to read more about NSP14, if I want to have now concentrate on, because I need a topic for the next week. So I want to concentrate now on NSP14.
Okay, I have a very simple paper that talks about the functions of the different coronavirus genome proteins. I will send that to your team, as well as we can start with that. If there's more needed, I will obviously share those with you.
I know, because if we really want to find out something, we need to now start working on, when we are talking about mutations, then do we really need drugs like overuse of drugs like antivirals after, because after the second or third day, because if the virus is not living after 7 days, patient has recovered. Why continue remdesivir for 10 days? Why continue favipiravir for 14 days when we know the virus is dead on day 7?
So this situation I think is unique with so many accumulated mutations because it is a combination of both antiviral failure, not failure, effect of antiviral and failure of the immune system. So this has escaped. It's simply a matter of selective pressure that allowed this mutation, for this virus to survive and escape.
What about plasma therapy? When we give plasma therapy, how does it work on NSP14?
Plasma therapy does not work on NSP14. Plasma therapy simply takes antibodies from a survivor and gives it to a patient that is having an active infection. So whatever virus is found in the bloodstream is theoretically neutralized with the antibody from the patient who has donated the plasma.
लेकिन इसका कोई ताल्लुकात नहीं है as far as the mutations are concerned, because there is one theory.
It does not affect the virus mutation in any way. It does not have any selective pressure on the virus replication.
Okay, so normally, when we had 614G mutation, which is in a non-RBD domain of the spike protein, which also had effect on neutralizing antibodies, 1B, and which also has increased transmissibility, but there was no hue and cry. 614G, there was no hue and cry.
There was some, because I think it came to Gujarat and people were very upset about this.
It came to Malaysia. It started with Malaysia. Then people said one of the strain has gone through India to Malaysia, through such and such place. But it never created. Is it the vaccine? Because of the vaccine, hue and cry is there? Because 614G never created a hue and cry.
No, I see. What happens is different. Nowhere in the world are you going to get the exact same mutations, because different selective pressures, and it's sort of like a deck of cards. You don't know what mutation is going to come where in the world. each of us could be under pressure with the same factors and we could still be reacting differently. That's precisely what has happened. The 614 mutation, maybe it didn't transmit very effectively. There was a hue and cry about 614 being more lethal. Maybe it was more lethal, but it didn't transmit very effectively.
So I want to come back to one more thing, is that it's a common sense that after 9 months of WhatsApp diploma being done by everybody in the world, everybody has done a WhatsApp diploma on corona prevention.
Yes.
Okay. So it is very difficult to understand that the second wave, if it is larger, bigger, and earlier than the first wave, if the second wave is larger, broader, earlier than the first wave, and for every second wave, there are two reasons: human behavior or a virus behavior.
It might be a combination of two, because
Yes. So therefore, whenever there is a second wave which is bigger, broader, and larger than the first wave, one should be working on a possibility of a mutation. Then why did UK missed it for 3 months? They are now
because we
tracing
don't
it
know
back
what
to September.
See, when you're focused on disease control efforts, surveillance is important, but when you're focused more on disease control, surveillance takes a second backseat. And then it's simply a matter of allocating your resources on where you can get most effect. Well, let us say this particular funding or whatever is better spent on controlling the infection rather than going back and sequencing so many genomes. When you see
No,
the effect,
I still want to say is cluster 5 of Denmark. Cluster 5, which came in the Minx, had 6970 deletion.
yeah,
And there was a lockdown in Denmark at that particular moment. And then they said in November that strain is extinct now. So there
right.
was a fear. So there was already a fear of 6970. Why did UK missed it? Because the same 6970 has carried on to now to 501. 501 independently also has been there in the previous times. So they have now combined together 501, 6970. So there was an alert by cluster 5 in Denmark, and there was a 501 is not a new mutation. It has
Yeah.
been seen.
It's
And
a,
in,
it's a combination. It's
and in the
always a combination.
experiment,
It's never
and
one
then
single mutation that makes the difference between a completely safe and virus, mild strain to a completely like a lethal variant. It's never one that makes a difference. It's the whole combination of different factors that make the difference always.
so that comes to our end. For those people who are on the Facebook and who want to understand what we have transpired, I like to. Do you like to finish it up in Hindi?
Me?
Yes, sum up in Hindi for the people who are on Facebook, or I do it.
No, you. I would request you to do it because my Hindi is very school book and I can understand where to get
So
water from, where is the bathroom. That's all.
पवित्रा ने बड़े सिंपल ढंग से लोगों को कहा कि म्यूटेशंस हैं, होते रहेंगे। हमारे वायरस के अंदर में एक सिस्टम होता है जिसे बोलते हैं, क्योंकि ये वायरस जब हमारे शरीर में आता है, हमारी सेल की फोटोकॉपी मशीन को हाईजैक कर लेता है और अपनी प्लेट उससे एक्सचेंज कर देता है जिसके वहां पे कॉपीज बननी है। क्योंकि इसमें एक प्रूफ रीडिंग मैकेनिज्म होता है, अगर वो प्रूफ रीडिंग मैकेनिज्म किसी कारण से खराब हो जाए, गलतियां करने लग जाए, जरूरत से ज्यादा गलती करे, तो म्यूटेशंस हो जाते हैं। अब जैसे इनका नाम मैंने पवित्रा लिखा है, अगर मैं वी के आगे डबल आई लगा दूं तो पवित्रा चेंज नहीं हो जाएगा, लेकिन म्यूटेशन हो जाएगा। आखिरी में ए के बदले 2 ए लगा दूं तो भी लोग इनको वही पवित्रा समझेंगे। सो देयर आर सिनोनिमस एंड नॉन सिनोनिमस टाइप्स ऑफ म्यूटेशंस। वो म्यूटेशन जो आपको आंख काले के बदले नीली कर दें, और वो जो आपका एक हाथ गायब कर दे जब आप पैदा हो, तो वो अलग अलग टाइप के म्यूटेशंस होते हैं। सो ये जो म्यूटेशन इस वक्त में हो रहे हैं, ये हो रहे थे, कुछ डेनमार्क में हुए, कुछ साउथ अफ्रीका में हुए, कुछ इंग्लैंड में हुए। 3 इंपॉर्टेंट हैं, एक 614G जो इस वक्त में इंडिया में सबसे ज्यादा है, एक याद रखिए न्यूयॉर्क 501 रुपए का, क्योंकि क्रिसमस है आज, तो 501 रुपए का आपने सबको शगन देना है। तो न्यूयॉर्क N501Y, न्यूयॉर्क का N, Y न्यूयॉर्क का, 501 रुपए का शगन, तो N501Y ये आपको याद रहेगा उसके साथ में। एंड 6970 डिलीशन आपको इसलिए याद रहेगा क्योंकि 269 और 270 आईपीसी के अंदर में अगर आप बिना मास्क पहने बाहर जाते हो और किसी को आप बीमारी फैला देते हो तो आपको जेल हो सकती है। तो 69 और 70 आप सब लोगों को आईपीसी के 2 ये कोड याद हैं। तो इस तरीके से आपको याद रहेंगे कि मल्टीपल म्यूटेशंस आपस में मिल गए हैं, और कोई भी ऐसी चीज जो मेरी मशीन के अंदर में NSP14 नॉन स्ट्रक्चरल प्रोटीन 14 नंबर को आपको याद रहेगा। 14 नवंबर को आपको चाचा नेहरू का बर्थडे है तो 14 नंबर आपको याद रहेगा। बच्चों के लिए 14 नंबर याद रहेगा कि 14 नंबर का प्रोटीन कोई भी बिगाड़ दे। अभी तो हम सिर्फ RDRP इनहिबिटर्स की बात कर रहे हैं, आगे जाके हो सकता है हमें कुछ और मिल जाए कि जो उसको सुधार दे या बिगाड़ दे, तो उससे म्यूटेशंस बन सकते हैं। जब तक ये म्यूटेशन मेरी डायग्नोस्टिक लैब को चेंज ना करे, अभी तो वो S जीन में कितना फर्क पड़ेगा पता नहीं, मेरे वैक्सीन को चेंज ना करे, मेरी बीमारी ज्यादा खतरनाक ना बनाए, मेरी बीमारी के अंदर में एंटीबॉडीज को काम ना करने दे, मेरी बॉडी में मेरी FIR लॉज ना होने दे, जिसे बोलते हैं सेलुलर मेमोरी सेल्स ना बनने दे, मेरी बॉडी में अगर वो ह्यूमोरल एंटीबॉडीज ना बनने दे। यस, इस सिचुएशन के अंदर में यू आर वरीड, अदरवाइज यू आर नॉट वरीड। सो दैट्स ऑल फॉर टुडे। लेट मी थैंक Dr. Pavitra to be here and be with us, and she has a nature's call with the boy, and so she had to leave. So let me thank you, Pavitra, to be here with us, and we'll be back tomorrow 7 p.m. with another show. Keep watching Med Talks with Dr. KK Agarwal. Namaskar, stay tuned.